Welcome to this explainer. Today, we're standing right at the fascinating intersection of physics and biology. We're going to look at how scientists are using polymers, the basic building blocks of so many materials, as these highly sensitive instruments to actually understand chaotic living systems. It is an incredible story about basically taking the temperature of chaos. Let's get into it.
Okay, let's dive into this. Here's our roadmap for today. We'll start with taking the temperature of chaos, then look at the polymer as a probe. From there, macro probing with living sludge worms, micro probing in the active bath, reading the spectral signatures, and finally, the future of active probes.
Section 1. Taking the temperature of chaos.
Seriously, I want you to think about this for a second. How do you actually take the temperature of chaos? We're talking about active matter here. These are environments like dense bacterial swarms or the highly dynamic, constantly churning interiors of living cells. In these systems, energy is being consumed and generated at the microscopic level by individual agents. The standard rules of physics, the ones designed for closed, quiet, boring systems, they completely break down here.
Now, what's really interesting is the stark contrast between these two worlds. On one hand, you have your standard equilibrium system. It's predictable, right? It has a single, universal temperature that tells you how much thermal energy is in the system, and it obeys the fluctuation dissipation theorem. Nice and tidy. But on the other hand, we have active matter. This system is microscopically fueled. It violates standard thermodynamics because it isn't just passively sitting there, it's pushing, pulling, and moving entirely on its own. A regular thermometer, it just can't capture that chaotic, non-equilibrium energy. We desperately need a new kind of probe to measure these active systems.
Which brings us to Section 2, the polymer as a probe.
Before we drop our probe into all that chaos, we need to understand its physical limits. In molecular physics, we use a vital measurement called contour length. Now, this is simply the length of a polymer chain at its absolute maximum physically possible extension. Think of it like pulling a curled-up piece of string perfectly taut. We absolutely have to know this baseline parameter because, as we're going to see, the active, chaotic environment is going to bend, twist, and deform this polymer. By knowing the contour length, scientists have a fundamental yardstick to measure all that deformation against.
And here is the key takeaway. When you place a semi-flexible polymer into a chaotic, active environment, what we call an active bath, it physically listens to the chaos. Because the polymer is long and flexible, it has different parts that wiggle and relax at entirely different speeds. It acts as a multi-scale probe, taking the chaotic pushing and shoving of its environment and filtering it right through its own physical shape. It literally gathers data through how it bends and moves. It's brilliant.
Moving right along to Section 3, macro probing with living sludge worms.
Okay, this is where it gets really fun. Scientists use living tubifolts worms. They actually act as macroscopic, active polymers with a contour length of about 15 millimeters. They put these worms into a flow channel filled with a hexagonal pillar array. Now, that's a classic chromatographic setup usually used for separating passive molecules, not living worms. As the flow pushes the worms through the pillars, scientists track their trajectories. And the really clever part? They can turn the activity of these living worms up or down simply by changing the temperature of the water. Talk about a biological thermostat.
So, the crucial point is how the worm's activity changes the way it navigates. When the water is a chilly 5 degrees Celsius, the worm is sluggish. It has very low activity, and it takes a really long time to get through that pillar maze, giving it a longer elution time. But bump that water up to 25 degrees, the worm becomes highly active. It wriggles frantically, constantly changing its conformation, and it actually navigates the obstacles much faster. This brilliantly proves that the physical conformation of an active polymer directly controls how it interacts with its environment. Mind-blowing, right?
Let's zoom in. Section 4, microprobing in the active bath. Down on the microscopic scale, the chaotic environment is made up of self-propelled particles. We call these active Brownian particles. They aren't just vibrating randomly due to heat. They actually have their own tiny internal engines. They're constantly colliding with, pushing against, and sliding past the polymer. Basically, they create a relentless active bath of microscopic forces that the polymer has to constantly react to.
Now, let's look at the anatomy of our microscopic probe. The polymer here is modeled as a series of connected beads. Think of these individual beads as tiny sensors. As those active, self-propelled particles crash into the chain, these beads experience noise. But here's the catch. It is not random noise. It's temporally persistent and spatially correlated. Because the active particles push in a specific direction for a bit before eventually turning, the polymer is subjected to a very specific sustained type of structural stress.
All right, Section 5, reading the spectral signatures. This perfectly illustrates how we actually read that stress. When you expose a polymer to the active bath, it doesn't just heat up uniformly all over. Instead, the activity specifically targets certain types of movement. The active forces dramatically amplify what we call the low-mode sector. These are the long, wavelength, slow, sweeping wiggles of the entire polymer chain. Meanwhile, the high-mode sector, which are the fast, short, jittery wiggles between individual beads, well, that remains mostly passive. It's largely unaffected by the active bath.
You can literally think of the polymer as a biological tuning fork. It doesn't just vibrate randomly. Its different shape modes vibrate right in tune with the specific activity level and the persistence time of its environment. The slow, persistent pushes of the active bath resonate perfectly with the slow, sweeping modes of the polymer, transferring energy selectively. So by reading which modes are amplified, scientists can actually map the properties of that invisible active bath. It's like reading the ocean's currents just by watching how a piece of seaweed sways.
Finally, Section 6, the future of active probes.
Because of this research, we basically have to completely rewire how we think about temperature in biology. It is a total game changer. We are moving away from the old idea of a single universal temperature for a living system. Instead, because our polymer probe absorbs energy differently depending on its wavelength, we have to look at an entire spectrum of mode-dependent effective temperatures. The temperature effectively changes depending on the scale you are looking at. How wild is that?
I want to leave you with this final provocative thought. If a simple, flexible polymer, whether that's a sludge worm in a maze or a microscopic chain in a computer simulation, can act as a sophisticated probe to read the chaos of active microscopic life, what other invisible engines could we map inside the human body? The tools we are developing today are quite literally giving us an entirely new language to understand the physics of life itself. Thank you so much for exploring this with me today. I'll see you next time.